Hello and welcome everybody to my channel Omni Bio Entrance, and today we will be discussing University of Hyderabad Integrated MSc PhD Entrance Examination for the subject Biochemistry and Molecular Biology conducted in the year 2020, and I will be discussing only the biology part. So coming to the first question. In pea plants, tall is dominant over dwarf. In a cross-pollinated cross, when 217 progeny were obtained, 103 plants were tall and the rest were dwarf. The genotype of the parents is most likely to be. So the total progeny is 217, and out of that 103 were tall and the rest were dwarf. So the number of dwarf will be 217 minus 103, that is 114. Now divide each of these, that is the number of tall and the number of dwarf, by the total number of progeny to get the ratio. So 103 divided by 217 is 0.47, that is approximately 0.5, and 114 divided by 217 is 0.52. So the ratio we see is approximately 1:1, that is half tall is to half dwarf. So when will you get half tall is to half dwarf? When you cross a heterozygous individual with a homozygous recessive parent, then you will get half tall and half dwarf. So the genotypes of the parents is half tall. That is the genotype of the parent should be capital T small t and small t small t. Then next one. When two tall pea plants were crossed, one obtained 75% tall and 25% were short. The genotype of the parent is most likely to be. So this is the 75% tall and 25% dwarf. That can be obtained when the genotype of the parent is both heterozygous for the tall plant. So capital T small t into capital T small t. So you will get 3 tall is to 1 dwarf. That will be 75% tall and 25% short. So the answer is option B, that is both heterozygous. Then next one. According to Michaelis-Menten equation of enzyme kinetics, what is the V0 by Vmax ratio when concentration of substrate is equal to 5 times Km? So we have the formula V is equal to Vmax concentration of S by Km plus concentration of S. Now S is equal to 5 times Km. When you substitute the value of S into this particular equation, you will get 5 Km by Km plus 5 Km, that is equal to 5 by 6. So the answer is option C, that is 5 by 6. The next one. Find the mean, median, mode respectively of these following numbers. So how will you find the mean? Mean is total sum of the numbers divided by total numbers. So this is the sum of the numbers, the total numbers. There are 13 numbers. So 52 divided by 13 is equal to 4. So mean is equal to 4. Then median. Median, you have odd numbers here. Odd number of numbers here. That is 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13. So 13 numbers are there. So the middle number will be the median. So first you have to arrange the numbers according to ascending or descending order. Then you have to see for the middle value. Then the middle value here is 5. So median is 5. Then mode. Mode is the value that appears most frequently in the data set. So here first arrange them into ascending or descending order and then see for the value which has got the maximum frequency. So 5 is having the maximum frequency. So mode is 5. So the answer is 455. Answer is option C. The next one. Consider Michelson stall experiment if E. coli cultures grown overnight in N15 ammonium chloride medium is shifted to N14 ammonium chloride medium and further grown for 1.5 generation time. What would be the relative proportions of N15, 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 N14, N14, N14 DNA at the end of the experiment? So you have got the N15 DNA that upon replication, N15 DNA upon replication will give N15, N15 only. Okay, now let that wait. Now we will take for N15, we will transfer them into N14 medium. So by semi-conservative method of replication, half will be N15 and the other half will be N14. So N14, N15, N15, N14, N15, N14, N15, N14. So then again you allow them to grow for the next generation. Then also you will get N15, N14, then N14, N14, N15, N14, 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 N15, N14, 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 N15, N14, N14, N14. By seeing this, you will see that there is no N15, N15 hybrid. There is no N15, N15 hybrid, so that will be 0%. Now, which is the maximum one? N14. N14 is the maximum one. So that will have more proportion. Okay. And which is the next one after N14, N14? It is the hybrid one. That is N15, N14. That is only appearing very less in number than N14 DNA. So your ratio should be C, that is 0% N15 N15, 33.33% N15 N14, because that is lesser in number than N14 DNA. But if you take this ratio, that is option B, if you see, you will see that they have written N15 N14 as more in number than N14 N14, but that is not correct. The correct answer is 0% and N15 N14 is lesser than N14 N14. So it will be 33.33% and 66.66%. So next one. In eukaryotic protein synthesis occurs in. So eukaryotic protein synthesis occurs in cytoplasm, mitochondria and chloroplast. So A, B and F are correct. Then given below are some and reactions that take place either in RBCs in systemic circulation, the tissue blood capillaries, or in RBCs in the pulmonary circulation, that is alveolar capillaries in lungs. Select the option that is not a correct match for the reaction and the site of the reaction. So outward the answer is option B, that is outward diffusion of chloride ions causes inward diffusion of HCO3 minus in RBCs from tissues in the systemic circulation. That is option B. Then a reaction buffer apart from substrate required the following composition for an enzyme activity. You are given 3 stock solutions. So these are the stock solutions. How much volume from each stock solution will you mix to prepare 10 ml of reaction buffer? So you are given the stock solutions as 2 molar tris at pH 9.5 and you need 200 millimolar of tris HCl at pH 9.5. Then you have got 0.5 mgCl2 as stock and you need to prepare 50 millimolar mgCl2. Then you have 0.1 molar NaCl as stock and you need 10 millimolar NaCl. So 2 molar. So from 2 molar tris. So for preparing tris, we will take 2 molar tris. How much we have to take from that 2 molar? So X. That is equal to 200 into 10 raised to minus 3 molar, because the answers are in because this is in molar, we have to convert 200 millimolar to molar into 10. So that will give you the value of X as 1 ml of tris. Similarly for MgCl2 you will get 1 ml of MgCl2, and for NaCl also you will get 1 ml of 0.1 molar NaCl. So you have to take combine and make up the volume to 10 ml. So the answer is 1 ml of tris at HCl at pH 9.5, 1 ml of 0.5 molar MgCl2 plus 1 ml of 0.1 molar NaCl. So the answer here is option C. Then next one. 4 proteins of molecular weights 15, 29, 46, 70 kDa respectively are present in a mixture. Each of these proteins need to be separated and isolated from the mixture. Given this information, which of the following options would be your strategy of choice to separate and isolate each of these proteins? Mass spectroscopy, affinity chromatography, combination of anion and cation exchange chromatography, gel filtration chromatography. Since they have given only molecular weight as the data, you need gel filtration chromatography for separating out proteins of this different molecular weights. So the next one. Given are the kinetic plots of two isoenzymes, that is glucokinase and hexokinase. Which of the following statements can be made by studying the plots? At fasting glucose concentration, glucokinase will be able to phosphorylate glucose at a higher rate than hexokinase. Because hexokinase has got lower Km value than glucokinase. So lower Km value means it has got high affinity for glucose. Okay, so hexokinase has got lower Km value means it will reach its half capacity at lower concentration than glucokinase. So under fasting condition, glucose levels are low. So hexokinase will be more active than glucokinase. So A is not correct. Then glucokinase has higher affinity for glucose. No, it has got lower affinity for glucose when compared to hexokinase. Then hexokinase reaches its maximum velocity only upon increasing blood glucose beyond fasting concentration. So that cannot be made out. Then next one. Glucokinase has lower affinity for glucose than hexokinase and its enzyme activity varies with glucose concentration. So that is the correct answer. That is option D. Then next one. Purified immunoglobulin was either papain digested or trypsin digested and the digestion products were resolved by SDS phase under non-reducing condition. So non-reducing condition, so the products are not denatured. Okay. So the question is identify the lane which represent papain digested IgG. So the Fab fragment, it is a 50 kDa disulfide bonded antibody fragment because upon digestion with papain for IgG, you will get two different fragments. That is FAB dash, you will get two Fab fragments of FAB and one FC fragment. Now this Fab fragment that you obtained, it is 50 kDa disulfide bonded antibody fragment that retains its antigen binding capacity. So it is 50 kDa. So they have asked here which one represent the papain digested IgG. So 50 kDa is shown by lane 2. So the answer here is option B, that is lane 2. Then a researcher was studying the activity of an enzyme in cells. The following observations were made in the study. Treatment with chloroquine induced the activity of the enzyme. So chloroquine will induce or increase the enzyme activity. Then addition of RNA polymerase II inhibitor along with chloroquine did not induce activity. So if you add RNA polymerase II inhibitor along with this chloroquine, there will not be any enzyme activity. That is natural because RNA polymerase II, it is needed for synthesis of the protein. So when you are adding an inhibitor of RNA polymerase, there is no enzyme activity. Enzyme is not produced and Even if you add chloroquine, there is no use. Then addition of cycloheximide. Now cycloheximide, it is a inhibitor of protein synthesis and it inhibits translation elongation. Up to 2 hours after addition of chloroquine, so up to 2 hours, if you add cycloheximide after the addition of chloroquine, what they observed is reduced the activity of the enzyme. So even though chloroquine was present, cycloheximide reduced the activity of the enzyme but had no effect if added later. So if you add cycloheximide after 2 hours, it will not have any effect. So based on this observation, identify which one of the following statements cannot be made about the enzyme. The synthesis of enzyme from mRNA continues up to 2 hours. Now that is correct because after addition of cycloheximide. Now cycloheximide, I told you, it is an inhibitor of protein synthesis. Up to 2 hours after the addition of chloroquine, that is chloroquine is one which will induce the activity of the enzyme. So addition of cycloheximide up to 2 hours after addition of chloroquinol reduce the activity of the enzyme. At the same time, when cycloheximide was added 2 hours later, it had no effect. So up to 2 hours, mRNA is translated to produce enzyme. That you can deduce from that particular statement. That means mRNA is present at least up to 2 hours. And after that is after 2 hours, it is not present. That is why when cycloheximide is added after 2 hours, there is no effect. So the synthesis of enzyme from mRNA continues up to 2 hours. That is correct. Then addition of chloroquine probably induced the expression of the gene encoding the enzyme. That also is right. Because treatment with chloroquine induced the activity of the enzyme. So that is also correct. Then fourth one, the DNA is present, the RNA is present at least up to 2 hours. That I already explained just now. So that is also correct. So what cannot be made out about the enzyme is rapidly degraded. We cannot make out from the study. So the answer is option C. Then the major factors determining the flux of glucose oxidation by aerobic or anaerobic glycolysis is or are. So that is NADH and ATP-ADP ratio are the major factors determining whether glucose is oxidized by aerobic or anaerobic hydrolysis. So the answer here is NADH and low ADP. Answer is option D. Then next one. Correct order of the 4 events during mitosis of a normal cell are. So first one is replication of bulk DNA, separation of sister chromatids, replication of telomeric DNA, arrangement of the centromeres to spindle fibers. So the first one will be replication of the bulk DNA that take place in your S phase. Then next one will be replication of telomeric DNA, that is option 3. Then after that you have got arrangement of centromeres to spindle fiber, that is 4, and then separation of sister chromatids, that is 2. So the answer, which is the correct order during mitosis, is 1, 3, 4 and 2. Then next one. The fluidity of the plasma membrane increases with which of the following? So the fluidity increases in unsaturated fatty acids in the membrane, that is correct. Increases in glycolipid content in the membrane, increases in phospholipid content in the membrane. So which one, the fluidity of plasma membrane increases with increase in unsaturated fatty acids in the membrane. So the answer is option B, that is A only. Then if V is the variance and sigma is the standard deviation, then variance is equal to square of standard deviation. So V is equal to sigma square. Then which of the following statements about the nucleus is incorrect? Nucleus is surrounded by double membrane. Yes, nuclear pore complex supports bidirectional transport across the nuclear membrane. That is also correct. Because bidirectional trafficking occurs continuously between the cytosol and the nucleus. Then proton pumps in the nuclear membrane maintain intranuclear pH. So that is the wrong one, because proton pumps, they are located in plasma membrane, mitochondrial membrane and chloroplast thylakoid membrane. So that is wrong, incorrect. Nuclear envelope remains intact during mitosis in many fungi. That is also correct. So the answer is option C. Then biological nitrogen fixation requires. It requires nitrogenase enzyme complex. Answer is A. Which of the following are unifying features of cephalochordates, urochordates and vertebrates? So they have a hollow nerve cord dorsal to the notochord. Answer is option B. Then, which of the following combinations represent all negative interspecific interaction in an ecosystem? So commensalism and amensalism. Or it must represent all negative interspecific interaction. Parasitism and mutualism. Parasitism is negative, mutualism is positive. Predation and commensalism. So predation is negative, commensalism is positive. Competition and amensalism. So option C is correct. Now before that, amensalism, it is the association between organism of two different species, each one in which one is inhibited or destroyed and the other is unaffected. Whereas commensalism, it is the association between two organisms where one is benefited, where the other is neither harmed nor destroyed. So which are all negative is competition and amensalism, that is option 5. Then next one, Hamilton's rule, R B greater than C. So B is the benefit gained by the recipient of the altruism and C is the cost suffered by the donor while undertaking the altruistic behavior, and R is the genetic relatedness of the altruist to the beneficiary. Predicts that natural selection should favor altruistic act under certain circumstances. So which of the following circumstances are likely to lead to the selection favoring altruism? So as per Hamilton, natural selection favors genetic success, not reproductive success. So it recognizes that individuals can pass copies of their genes on to future generation through direct parentage, that is by rearing of offspring and grand offsprings, as well as indirectly by assisting the reproduction of close relatives such as nieces and nephews through altruistic behavior. So altruistic behavior is that behavior that benefits other individuals at the expense of the one performing the action. So now if you take one example, suppose there are two lion sisters, both have cubs. So the female lion has well nourished cub and her full sister has got a starving cub. Now what will the female lion do? She will start to nurse the starving cub of her sister. Now in addition, she gains inclusive fitness. So the female lion, she gains inclusive fitness on starting to rear the starved cub of her sister. So according to Hamilton, inclusive fitness refers to the ability of one's gene to survive both in one's own offspring and in one's genetic relatives. So by doing this altruistic behavior, she is sharing her genes, or the female lion is sharing her genes to the starving cub. So this means that female lion with a well-nourished cub gain inclusive fitness by nursing a starving cub of a full sister, because the benefit to her sister, that is B, that is benefit gained by the recipient of the altruism, is more than compensates for the loss to herself, that is by feeding the starving cub, the one who is doing this altruistic behavior, she is having a loss, but that loss, that is C, is the cost suffered by the donor while undertaking the altruistic behavior. So that C is the cost suffered by the female lion while she is in the act of protecting the starving cub of her sister. Okay, so that is C. So C is the cost suffered by the donor. So it more than compensates for the loss to herself. So the loss to herself is loss for the female lion. Now that is 1/4 of the offspring. Okay. Since the survival probability of her own non-starving cub is only slightly reduced, and the survival probability of her own, that is of the donor's non-starving cub, is only slightly reduced. Given that the average genetic relatedness, that is R is the genetic relatedness of the altruist to the beneficiary. So genetic relatedness between two full sisters is 0.5. Then according to Hamilton's rule, 0.5 into 1. So R into capital B is greater than 0.25, that is C. So in total what happens is that in essence the genes for altruism spread by promoting aid to copies of themselves. So R is the genetic relatedness of the altruist to the beneficiary, that is 0.5. So genetic relatedness of the altruist, that is the female lion, to the beneficiary is the starving cub of the sister. And B is the benefit gained by the recipient of altruism. So she is gaining benefit by sharing her genes with the starving cub of her sister. So that is 1. So benefit gained by the recipient of altruism is 1, and C is the cost suffered by the donor. So it is 1/4. 1/4 is suffered by the donor. So that is 0.25 while undertaking the altruistic behavior. So which of the following circumstances are likely to lead to selection favoring altruism? So when R is small and B is large, it will favor selection of altruism. So R is small but B is large. That is the answer. That is genetic relatedness of the altruist to the beneficiary is small but B is large. That is benefit gained by the recipient of altruism. When that is large, then circumstances are likely to lead to selection favoring altruism. So coming to the next one. Which of the phosphates in ATP is attached with the OH group of an amino acid in a protein when the latter is phosphorylated by its kinase? So it is gamma phosphate. Answer is option C. The next one. Tautomerization to the immunoform of cytosine would lead to transition. Now transition is, before that, due to cytosine tautomerization, it will pair with adenine. Okay, so cytosine tautomerization, it will pair with adenine, and in the next round of replication, adenine will pair with thymine. So cytosine and thymine here are pyrimidines, whereas adenine is purine. So first cytosine is converted to a purine, and the purine base pair with the thymidine. Thymidine, that is adenine base pair with thymine, which is a pyrimidine. So cytosine, a pyrimidine, is replaced by another pyrimidine, that is thymine. So the case of transition where one pyrimidine is replaced by another pyrimidine, or one purine is replaced by another purine. So answer here is option A, that is transition. The next one. Which of the following statements about ecological pyramid is incorrect? So pyramid of energy of freshwater ecosystem is inverted. So that itself is incorrect. Why? Because pyramid of energy can never be inverted. The next one. Which of the following is an example of second class liver functioning through our skeleton muscle systems in humans? So standing on tiptoes, that is metatarsophalangeal joints with the pulling force across the heel of the foot. So that is the answer. So answer is option B. Then which types of hybridization of the carbon atom are found in 2-butene? So 2-butene is CH triple bond CH. So you will find sp and sp3 hybridization. So answer is option C, that is sp and sp3. Name the carbohydrate structure given below. So it is beta D galactopyranose. So answer is option A, beta D galactopyranose. Then which of the following two amino acids can have four stereoisomers? So threonine has two chiral centers, therefore this is threonine. It has got how many? Two chiral centers. 1, 2. So, 4 possible stereoisomers. Then this is isoleucine. Isoleucine is also having two chiral centers. One here and one here. And so, therefore, 4 stereoisomers are possible. So, threonine and isoleucine are the answers. So, option C is the answer. Then next one, match the contents of the column A to the correct content of column B. So Darwin, survival of the fittest, 1 D. Then Malthus, Malthus's population tend to multiply more rapidly than food supply, 2 A. Then next comes Lamarck. Lamarck is inheritance of acquired characters as basis of evolution, 3 C. And De Vries is evolution progresses through discontinuous variation or mutations. So answer here is option B, that is 1D, 2A, 3C and 4B. Then hydrolysis of the bonds shown by arrows and numbered in ATP result in release of free energy. Choose the correct answer from the following option regarding the amount of free energy released by hydrolysis of these bonds. So if hydrolysis of 1 and 2 take place, you will get 7.3 kilocalories of energy. So 1 is equal to 2, but for breaking the third bond, you require more energy. So 1 is equal to 2 greater than 3. The answer is option C. Then which of the following statement is correct for anabolism and catabolism? Exergonic breakdown, endergonic synthesis. Answer here is endergonic synthesis and exergonic breakdown respectively. So endergonic reaction that requires free energy to proceed. So energy absorbed from the surrounding. And exergonic reaction releases energy to the surrounding. So endergonic synthesis and exergonic breakdown respectively. Then next one. Given below are symbols that are used in floral formula and their meaning. So one, see, that is epipetalous stamens. Epipetalous stamens. One C is epipetalous stamens, and fourth one, that is 4 is, sorry, 2, second one. Okay, 1 minute, I made a mistake. Okay, second one is A, that is hypogyny. Third one is epiphyllous stamens, and 5 is epigyny. So the answer is 1 D, that is 1C, 4A, 2A, 3D and 5B. Then formation of D-fructose 1,6-biphosphate from the combination of dihydroxyacetone phosphate and D-glyceraldehyde 3-phosphate is an example of which one of the following biological reactions? So it is an example of aldol condensation, that is C. Then next one. Which of the following organic compounds would be the most acidic? So NO2, C6H4OH, that is the most acidic. Then which of the following amino acid is considered as both ketogenic and glucogenic? So the class of amino acid that produces pyruvate or other glucose precursors during the catabolism of amino acid are called glucogenic amino acid. And the class of amino acid that produce acetyl-CoA and acetoacetyl-CoA during the catabolism of the amino acid are called as ketogenic amino acid. Now the amino acid which is both ketogenic and glucogenic is tryptophan. Answer is option A. Then mammalian cells are maintained in the flasks with liquid media and stored in an incubator with proper carbon dioxide levels. If flasks are taken out of the carbon dioxide incubator, the liquid media will show which of the following changes over time. So they will show an increase in the pH of the medium. Now a carbon dioxide incubator is used in cell culture to provide it with optimum temperature, moisture and to maintain optimum pH. So once you take it out, you get an increase in pH of the medium. And next one, which of the following pair of ions does not have incompletely filled d orbitals? Now Sc3 plus and Zn2 plus. Now Sc3 plus has got the electronic configuration 1s2, 2s2, 2p6, 3s2, 3p6. So it is all completely filled d orbitals. And Zn2 plus, it is having 1s2, 2s2, 2p6, 3s2, 3p6, 3d10. So that is also having completely filled d orbitals. So the answer is option C. Next one. A solution has concentration of H plus is equal to 7 into 10 raised to minus 7 molar. Which of the following is true for this solution? So minus log concentration of H plus will give you pH. So minus log 7 into 10 raised to minus 7 is equal to 6.154. So the solution is acidic. Answer is option B. Then glutamate dehydrogenase enzyme is present in. So it is present in cytosol and in mitochondria. Answer is option B. Then which of the following is not an enzyme catalyzed reaction in metabolism? So it is Maillard reaction. Now it is a non-enzymatic reaction between sugars and proteins that occurs upon heating and that produces browning of some foods such as meat and bread. So the answer here is option D, that is Maillard reaction. Next one. What is the number of proton NMR peaks observed for 1,3-dinitrobenzene? So it is 3 peaks are observed. Then when a G2 phase cell is fused with the M phase cell, which of the following events occur? So G2 phase, the cell grows more make proteins and organelles and begins to recognize its content, begin to reorganize its contents in preparation for mitosis. G2 phase ends when mitosis begins. And during the mitotic phase, the cell divides its copied DNA and cytoplasm to make 2 new cells. The nuclear DNA of the cell condenses into visible chromosomes and is pulled apart by the mitotic spindle. So when you fuse G2 phase and M phase cell, which of the following events occur is there can be gene transcription. Answer is option A, as well as premature chromosome compaction. So that is both A and C are correct. An open reading frame is a protein coding region in DNA. So answer is option B. Then next one. E. coli with mutation in LAC-Y gene was isolated. This strain was grown in medium containing glucose and lactose. Which of the following graphs correctly represent the most likely growth pattern of this mutant E. coli? So it lacks Y and sorry. So this particular E. coli, it has got mutation in the LAC-Y gene, and LAC-Y it code for permease enzyme. That is, it facilitates and regulates permeability of lactose into the cells. So once glucose has been utilized, the E. coli will not be able to utilize lactose. So it will be option C will be the answer. Then the -10 box sequence of the gene that is transcribed by sigma 70 in E. coli is 5' TCTAAT3 3'. Which one of the following changes is likely to increase transcription maximally from this promoter? So if you have this particular sequence, that is option D, that is TAT AAT, because that sigma 70 it recognizes the consensus minus 10 TAT AAT sequence. So if you have that particular sequence, it will increase transcription maximally. A cation exchange resin was used to separate a mixture of charged proteins. All of the following statements about the experiment are correct except proteins that carry negative charge in the experimental condition will flow through and are not retained. Strongly positive charged proteins will require higher ionic strength buffer to elute. Larger molecular weight proteins will be eluted first. So, and uncharged proteins will not bind to the column. So, cation exchange chromatography, it more specifically uses a negatively charged ion exchange resin with an affinity for molecules having a net positive charge. So here this option C, that is larger molecular weight proteins will be eluted first, is not possible. It is possible with gel filtration chromatography, because cation exchange resin it separates protein based on their charge, not on the molecular weight basis. So answer will be option C. Then next one. Which of the following is not generally a part of eukaryotic transcription? Capping of the 5' end of the mRNA with methyl guanine. That is correct. Multiple initiation and termination codons per RNA molecule. That is not a eukaryotic transcription mechanism. Then addition of non-templated string of poly A, removal of intervening sequence and splicing of the exon. The A, C and D are correct, whereas D is not correct. So thank you for watching my channel. Please like, share and subscribe. Thank you.